The goal of the present study was to quantify the levels of cell death following different hypothermia treatments in a human cardiomyocyte-based model. This model can be used to study the molecular mechanism of hypothermic cardioprotection, which may have important implications for the development of complementary therapies for use with hypothermia. To culture the HCM cells, thaw the cryopreserved cells from liquid nitrogen in a 37 degrees Celsius water bath and maintain them in a humidifier incubator under an atmosphere with 95% air and 5% carbon dioxide. Once the cells reach 60 to 70% confluence, aspirate the medium from the 6-well plate and gently wash the cells 3 times with PBS. Add 2 milliliters of fresh sugar-free medium into each well and culture the cells at a constant temperature in a 3-gas incubator under a mixture of 95% nitrogen, 5% carbon dioxide, and 0.1% oxygen at 37 degrees Celsius. For the time temperature protocol, aspirate the medium from the 6-well plate and add 2 milliliters of fresh sugar-free medium. Culture the cells in a tri-gas incubator for 12 hours to establish hypoxia. Set the temperature program, starting with 10 hours of low-temperature treatment, followed by a rewarming phase for 2 hours, and 24 hours of normothermia. Remove 3 petri dishes at